Macular Society London Conference, 2013. Professor Rachel Williams, Liverpool University. Engineering, Cell Transplantation and the Treatment of Macular Degeneration.
I'm actually an engineer. I'm not a clinician, and so a lot of what the clinicians do and what Claire has very beautifully explained is quite difficult for me to understand as well. And I'm learning exactly the same way as you are all the time about this. But I've worked with the ophthalmologists in Liverpool now for about 15 years. And over that time, what we've tried to do is to bring our thinking together. And maybe many of you in this room may be engineers or know people who are engineers. And in some ways, we have a slightly different way of thinking about things than the clinicians do. And by bringing the two approaches together, we can often solve the problems that perhaps seem intractable to one or other side. And that is what really excites me. It's the multidisciplinarity of trying to solve these problems and bringing the different approaches together. There are three things that I'm particularly interested in. So I'm an academic engineer, and so I'm trying to address the real issues underlying these problems that we can then take these on and deliberate them and translate them into clinical practice to really try and improve the situation for you guys. But the three areas I'm really interested in is in terms of the cell transplantation for treating AMD, age-related macular degeneration. And I'll talk to you about some of the areas of the research that we're doing in that area. I've also been involved in developing tamponade agents, which are used for vitreous replacements, and trying to put an engineering approach onto how we can improve the materials to make them work better, so that if you have to have a vitrectomy where they remove the vitreous from your eye, as Claire was explaining, then we can replace it with a material that's going to behave really well, so you get a long-term good clinical outcome from it. And I'm also interested in intraocular lenses. Those of you that have had to have vitreous replacements will also probably have to have an intraocular lens, and those can end up having a problem with post-operative scarring. And if there are ways we can improve those materials so that we can reduce that as a problem. So I may not have time to go into the intraocular lens one in detail, but I'll try and cover the other two. And one of the things that also Helen asked me to do was to talk about some of the sort of gene therapy areas that are going on. This is not an area that is my area of research, and I've done a lot of reading though about it, and I'll try and sort of update you on some of the work that's going on in that area. But it's a little far from my comfort zone, if you like, so don't ask me too many awkward questions on it, certainly. I'll struggle with that. Let's start by talking about the cell transplantation therapy, and Claire mentioned stem cells. I think just as a side to that, it's not necessary that we have to use stem cells. And in fact, our approach in Liverpool is not to go down the stem cell route. And what I'll do is I'll try and explain to you what our strategy is, if you like, and how that compares with the London project, which I'm sure a lot of you know about, where they are developing a stem cell strategy. But when you're trying to look at this stem cell therapy, what we're doing is we're looking at the cells in the eye that are problematic in age-related macular degeneration. And as Claire pointed out, these predominantly, what we're talking about here is the retinal pigment epithelium, the layer of cells underneath the retina, sitting on top of the choroid and on the Bruch's membrane. And it's these cells that become influenced and degraded by the build-up of drusen, as Claire explained. That the role of the cells is fantastic. You follow somebody who's done a lot of the work for you, so she's already told you how these work. They're important layer of cells that deal with the nutrients to the retina and getting rid of the waste from the retina. And if they don't do that, they begin to degrade because they get this layer of waste products underneath them. And it's what we want to do, therefore, in the cell replacement therapy is to put in a new set of healthy cells and get rid of these degenerated cells. And there's proof of concept that this has the potential to work. There's a very complicated surgery that can be done where they take what's called a patch graft. They take some of your peripheral RPE cells, retinal pigment epithelial cells, and they take them from the periphery and they move them under your macula. and have demonstrated that in certain cases that can improve sight in certain groups of patients. It's a very complex surgery, and also you could end up with quite a lot of scar tissue associated with it. So it's not something that's going to be readily available for a lot of people. But it does provide us with proof of concept that if we do replace these degenerated cells with healthy cells, then you have the potential to improve sight. So our approach is to say, well, we don't really want to take cells from your periphery. You've got some cells there that are healthy and working. We don't want to waste those. You might need to use them. So can we actually replace them with healthy cells from somewhere else? To be able to do this, there are three things we need to be able to do. We need to grow them on something. We need to grow them on a membrane that we can easily get hold of, something that we can use in the laboratory. We need to get some cells from somewhere, stem cells or maybe cells from the same patient. And we need to demonstrate or work out the surgical procedure that will actually allow us to get this membrane plus its new healthy cells back into the eye. And all of these have got to come together so that we can actually develop a new procedure that's appropriate and usable by the clinicians and to help you as a new treatment. Now, the bit of it that I'm particularly involved with from my engineering perspective is the membrane. And what we need to do that is we need a material. It's got to be very, very thin. It's got to be around the thickness of 1/100th of a millimetre. So a very, very thin layer of material. It's got to be porous, because we've got to get the nutrients through it and the waste products back through it again afterwards. It's got to be flexible and handleable so that the surgeon can get it into the back of your eye and lay it down to the macula underneath your retina. And it's got to have very good, what we call biocompatibility. It's got to interact with the cells such that we can get a monolayer of these cells growing on this substrate, and then it's got to protect those cells and look after them once it's put back in the eye. You'd think that's not too bad, maybe we can do that. That's quite difficult, we've got a lot of things to look at. Now, actually, we've got two materials, though, that work very well. And one of these materials you all know. It's a material, and I'm going to tell you its name because you'll all say, oh gosh, it's called Gore-Tex. Now, you all know what Gore-Tex is, don't you? You all come across it in your waterproofs. Now, Gore-Tex is actually a material that's made out of a polymer called polytetrafluoroethylene. You don't need to know that, but it's also, that is a material you're also very familiar with. It's Teflon. Now, Teflon is all used on your non-stick saucepans. This is a material that is very well known, and it has many that, in the form of Gore-Tex, it's porous. It can be made in very, very thin films and very, very thin membranes. It's very biostable, so it doesn't break down in the body, and it's actually already used in medical applications in the cardiovascular area. It's used as a film to mend holes in the heart if you've got a fissure between two of the chambers in your heart. So it's also a material that's already well used to being used in medical practice, which is a big advantage for us. Now, one of the things, what about Gore-Tex? You use Gore-Tex in your waterproofs because it doesn't like water. It's a material, we call it hydrophobic. Hydro meaning water, phobic meaning hating. And in fact, you also know that if you took your saucepan or your frying pan, you put a drop of water on it, it beads up in a really nice tight little droplet. That's because the water doesn't like to make contact with it. It's water-hating, hydrophobic. Now, unfortunately, cells don't like to grow on it either because it's hydrophobic. So what we do in the laboratories, we've got a really nice, simple process. It's called a gas plasma process. And it's a process which you can put your material into a chamber. You may have seen these a while ago. They're sort of things that you have in science museums. You have a globe, and in there, if you put your hands on it, you get a sort of like a lightning strike within it. That's a gas plasma, and it's a highly charged gas. If we put our material into that gas, it actually breaks the bonds in the surface of the material. We take it out and put it into water. The water interacts with that surface, and we now have a material that's exactly the same properties as the base material in terms of the bulk, but the surface layer, the minuscule, the atomically surface layer has been modified such that we've now incorporated functional groups into that surface that make it hydrophilic, water-loving. So we now have a material that's porous, it's very thin, it's nice and flexible, It's very biostable, and we've now got it so that in the laboratory we can grow a beautiful monolayer of retinal pigment epithelial cells on it, which are now ready for transplantation back into the eye. Works beautifully in the laboratory. You know what's coming next. The other side of it now is we have to decide what cells we're going to use. Now, our strategy in Liverpool is that not to go down the stem cell route. I'll compare it with the stem cell route in a minute. We think, though, there's an advantage of using the patient's own cells in terms of immune response. If you use cells from somewhere else, you potentially have immune response, and you therefore need to have anti-rejection drugs when you put these other cells into your eye. But there's a potential, we don't want to use retinal pigment epithelial cells because we don't want to use any of your healthy ones that you're going to need. You have, though, cells in your iris called iris pigment epithelial cells in the coloured part of your eye, and they actually are very, very similar to retinal pigment epithelial cells. It's very easy to get to your iris, and in fact, there's a procedure in treatment of glaucoma is to take an iris snip and make a small hole in your iris. And so these cells can be harvested very easily from that patient. So our route would be to take these cells from the iris and put them onto our substrate in the laboratory, grow them up into a monolayer, and then implant them as a monolayer of functioning cells on our substrate into the back of the eye, into the same patient. And we believe that that overcomes a lot of the issues of using the stem cell route. We can do this in the laboratory. We've done a lot of work, again in the laboratory, in fact using pig eyes, enucleated pig eyes, but we have some human eyes that are donated for research as well. And we can grow IPE cells, iris pigment epithelial cells, on our substrate, and they behave in a very similar way to the retinal pigment epithelial cells on the substrate. So the next stage is to develop the procedure. And we work very closely with the vitreoretinal surgeons in Liverpool. And we have a mock operating theatre in our laboratories. And again, we use our pig eyes that we just get from the abattoir. And we've demonstrated that we're working out how we can develop an instrument so that we can hold our very, very thin membrane. Remember, this is 1/100th of a millimetre thick, with our cells already on there, which are very delicate, and how we can then put that down into the eye through the limbal region of the eye into the back of the eye. At the same time, they have done a peripheral retinectomy, moved the retina out the way, put the implant down onto the macula, and then put the retina back on top, and then replace the vitreous with a tamponade agent. And it all sounds very simple, doesn't it? And this is something that we're practicing in the laboratory at the moment. We've developed an instrument with our colleagues in the engineering department where we've drilled little tiny holes into needles using lasers so that we can hold the substrate onto the shaft, if you like, of the needle with gentle suction. And then when we've got it into the back of the eye, we change the suction into a flow of fluid, and it floats it off down into position. Technically very difficult, but it's working very well. The situation we're at the moment is this is all working beautifully in the laboratory, and we need to move it on to the next stage. And we need to do that, we need to do an animal study. Unfortunately, we can't go straight to clinical trials with these things. And to prove that once we've got our membrane in position, that we don't cause any scarring or inflammatory responses that are going to cause long-term problems. But we're looking at the moment for funding to get to that next stage. Now, let me just briefly, as I said, I would compare this with the London project. The London project is doing very similar things, and we work very closely. There is a lot of collaboration between different groups on this, and definitely internationally as well. But I think, in fact, in many of these areas, the UK is very much at the forefront of some of this work. In the London project, they are using different substrates to us. I think they'd quite like to use our substrates, actually, and I think that's something that we're going to try and move on to collaborating, because I think we've done more development of the substrates. Their approach is to use embryonic stem cells. Now, embryonic stem cells are a source of cells that are readily available. And that's an advantage because you don't have to collect them from the individual patient, and the patient doesn't then have to wait while those grow up in the laboratory before you put them back into the eye. The embryonic stem cells have some concerns still in terms of what's going to happen long term when they're put back in the eye. There are a number of clinical trials, and there's a group called the Advanced Cell Technologies group in California who've done some clinical trials injecting these cells into the back of the eye over a number of years now, and I should say that it's a safety trial, and there's been no problems in terms of the safety of these materials, these cells. They're only injecting them as a suspension, and I think both the London Project and us would be completely in agreement that if we're going to find a new strategy for treating AMD, we need to be implanting these cells as a functional monolayer from the outset. The injection of a suspension of cells is not going to be functionally a strategy that is appropriate. But the safety trial is the only way that we can actually test this out. The London Project is going through the regulatory approval process at the moment so that they can start a clinical trial of the embryonic stem cells on the substrate they're looking at. And when I last spoke to Professor Coffey, they were still hoping to start that within the next few months. I think on their website they say within 2013. But then there is not yet a list, unfortunately, that anyone can sign up to. But I think it's a question that should hopefully be coming quite soon. So it's very encouraging. I think we are getting to the stage now where by bringing the engineering approach together with the science and the cell biologists, we're getting to the stage now where we're understanding a lot more and have the potential to come up with a strategy that might be going to work for this area. And it's really a question of timing. We've got to do the really good science to demonstrate that it's going to work before we start to actually use it on patients. Let me move on now and have just briefly about tamponade agents, because if we do this procedure, we're going to have to replace the vitreous and put a tamponade agent in there to hold down the replacement membrane with its cells on it underneath the retina. And you may know that the tamponade agents that are used are usually silicone oil. And it does sound a bit odd to be putting an oil in the back of your eye. But I think, and I'm sure Claire would agree, that these actually have been used for very many years now, and they're very successful clinically. But they do have some problems. And one of the problems is that they tend to emulsify. Now, if you can imagine you've got an oil, if you're making your French dressing, salad dressing, you take an oil, you take vinegar, and you shake it up, and you get an emulsion. Well, unfortunately, that's exactly what can happen to a smaller extent in the back of your eye. You put the oil into the back of your eye, and then you've got aqueous solution around that with proteins in it, which are a very good surfactant. And as you move your eyes around, there's a potential for emulsified droplets to form around the oil droplet. So what we've been doing is we've put our engineering hats on again, and we've looked to see is there a way in which we can modify the oil such that we reduce or increase its resistance to emulsification. And we do this by adding a very small amount of a very, very high molecular weight material. This is a technology that actually is not new. It's a technology that's known and is used in the aircraft industry, actually. If in fuel, which are oils that are used in aircraft, if they had a potential to form these little emulsified droplets, they become explosive. So to deal with that, the petrochemical industry have provided ways of putting these very long molecular weight molecules into the fuel, and then that stops that happening. So we thought, well, we could actually try and use this in the silicone oils. So we take our normal silicone oil that's used for treating tamponade agent, treating retinal detachments, and we add this exactly the same chemistry, but a very, very long molecular weight. And it makes the material slightly stringy. If you'll excuse the analogy, sometimes, you know, if you spit, it gets a bit stringy. That's because you've got really long molecular weight molecules within your spit. And it's the same principle. If you can imagine it, it's exactly the same that we're doing to the oil. We're just making it slightly stringy. This actually has been very successful in reducing emulsification, but it actually also has another advantage. It makes it much easier for the surgeons to inject it. Because it's stringy now, as they inject it and push it down a needle, the stringy bits all line up, and that makes the shear viscosity lower, and they can inject it and remove it from the eye more easily as well. And actually, the clinicians that I work with think that's much more important than the lack of emulsification, because it makes their life a lot easier. So you always find that when you're doing these experiments and you're addressing these problems, you need to talk to your clinicians and find out what really is the problem for them. This is quite nicely as a product that's now actually become onto the market. And in fact, in 2013, they brought out another one, a company that are actually selling this, and the new one should be even more emulsification resistant. So it's a way in which we brought our engineering knowledge to address this problem and see whether we can't actually, by bringing heads together, looking at a problem from completely different points of view, can actually try and bring a solution that can really benefit you people who are desperate in terms of having a problem with losing your sight, which we all know is so important. One other area that we're working with, in fact, with the tamponade agents is to make them heavy. If you remember, going back to the analogy of making your French dressing, if you put your oil and vinegar together, the oil always floats. Well, that's fine if your retinal detachment's in the top of your eye, but if the retinal detachment's in the bottom of your eye, that's not really very useful. So can we make the oil heavy? And we've developed a way of doing this by adding silica nanoparticles to the oil. So we're adding a solid. We're basically just adding sand. It's very clean, controlled sand, but we're basically adding sand to the silicon oil. And by doing some clever physics, where we refractive index match the oil and the sand, it's completely transparent. So it doesn't actually cause any problem in terms of the clouding of the oil. And we can add different amounts of sand and make it different amounts of heaviness, which then the surgeons can then choose which oil they would like to use in a particular situation. So again, it's another way where we've brought in together our knowledge of material science and how we can change these materials to try and make them work better for you and the clinicians. Now, I said I would mention the gene therapy. Are we all right for time? We'll quickly carry on very, very quickly. That gives me enough excuse to go through it very quickly. Gene therapy, as Claire said, is looking at some very interesting areas. In the problem with some of the macular degeneration diseases is that they have a genetic link to them. So there are cells in your eyes which are missing the particular genetic material. There's some very, very good work going on where they can try and replace the genetic material with synthetically produced genetic material using a gene transfer process of using viral vectors. And there's since about 2007, there's been some clinical trials going on where they have looked at the safety trials of injecting these gene vectors into underneath the retina and getting the genetic material incorporated into the cells and demonstrating that they are really very safe and are causing really no detrimental effect to these patients. Unfortunately, they're all safety trials, and there are very few that are actually looking at efficacy. And unfortunately, I think currently a lot of the data, particularly with Stargardt's and with wet AMD, that the improving in sight has not really been demonstrated yet. And the feeling about that is that it's a multi-component genetic disease, not just a one genetic disease. So we need to be replacing more than just one component to try and get it to work better. But it's very encouraging that there is the potential to do this and that the genetic engineering of it is really very, very successful. And I think this is something where there's real potential for in the next few years for there's some real progress to be made in actually addressing some of these issues. And I think this just brings me to my last point and why, if you compare what we're doing and what the London project are doing with a cell transplantation study and then some of these genetic studies, is that there's no one procedure that is going to be right for everybody. What we need to do is we need to have an arsenal of different approaches, because different patients or different solutions, different strategies will be better for one patient group than another patient group. And what we need to do as scientists is deliver and develop these strategies so that we can then work out which patient groups are going to benefit most effectively from which strategy. And I think the other thing that I would just like to warn you is that try and talk to the scientists and come to meetings like this, because I think the worry is that very often the press gets hold of some of these issues and they tend to hype it up. And it's very upsetting, I think, to find out or to think there's a solution just around the corner, but really it's a few years off yet. And I think what I could try and encourage you is to understand where we're coming from. We're working very hard to try and get these solutions, but they have to be right. There's no point in us putting a solution out that isn't going to work. And if we do that, and it gets tried on some patients and fails, it is then a very, very demoralizing process for not only you, but for us as well. So we are maybe a slightly cautious group, but we really want to make it work and not to have mistakes.